Hello everyone. Today we are going to discuss a case study regarding the isolation and purification of heparinase from Streptomyces variabilis MTCC 12266. So first let's have a brief introduction regarding the heparin and heparinase enzyme. Heparin is a sulfated polysaccharide composed of beta-1,4 linked glucuronic acid and N-acetyl glucosamine and other disaccharides. The depolymerization of heparin produces low molecular weight heparins, which have crucial biological roles and can act as therapeutic agents. Heparinase is an enzyme that brings about the enzymatic degradation of the heparin in the following manner. Initially, it damages the glycocalyx layer of the endothelial cells, and this damaged endothelial cells increases the impermeability of the blood vessels, which promotes the pre-thrombotic stage. Also, the damaged endothelial cells activates the platelets, which initiates the clot formation. Now let's discuss about the sample collection of the heparinase producing organism. The organism was isolated via the soil sample collected near the basins of the Gomti river, and it was further cultivated on a selective medium, namely heparinase selective medium, which contained heparin as a sole carbon and nitrogen source to indicate the hydrolyzing activity of the heparinase producing organism. After cultivating the organism, its growth rate was determined by cultivating it on separate mediums, namely heparinase selective medium, defined heparin minimal medium, and a complex medium of which one contained heparin and the other didn't contain heparin. After the incubation, it was observed that a medium which contained the heparin showed more heparinase production. So it can be concluded that the S variabilis showed an heparin dependent induction of the heparinase production. That is, it produces heparin more in the medium which contains heparin as the source. So now let's take a look at the different techniques used for the purification of heparinase enzyme from Streptomyces variabilis MTCC 12266. First, ammonium sulfate precipitation. Second, DEAE Cephadex A50 column chromatography, which is a type of anion exchange chromatography. Third, Cepharos D6B column chromatography, which is a gel filtration chromatography. And at last, its molecular weight determination by SDS phase electrophoresis. So now let's discuss each purification technique in detail. First, ammonium sulfate precipitation. To carry out this purification technique, initially the bacterial cell biomass is cultivated and collected by centrifugation at 4500 G. It is then stored in Tris buffer of 10 millimolar at pH of 7. The bacterial cell suspension is then disrupted by sonication through an ultrasonic processor for 10 minutes, and the cell debris are collected by centrifugation at 6000 G. Ammonium sulfate precipitation consists of two processes, salting in and salting out. Salting in. It refers to addition of salt of very low to low concentration, which helps in solubilization of the protein in the solvent. Salting out. It refers to addition of salt of high concentration, due to which the water now starts forming bond with the salt instead of the protein, and thus the protein remain alone and start forming aggregate of themselves, and this result in the precipitation of the protein. The precipitated proteins are then collected by centrifugation at 10,000 G. The pellet is dissolved in 0.01 molar phosphate buffer at pH of 7 and dialyzed against the same buffer overnight at 4 degrees Celsius. After the dialysis of the enzyme, an anion exchange chromatography, namely DAEE Cephadex A50 column chromatography of the enzyme is carried out. In this chromatography, Cephadex resin is used as the stationary phase, whereas the buffer solution is used as the mobile phase. So before discussing the principle of the chromatography, let's have a brief introduction to the components used in the chromatography. First, Cephadex. It is a range of cross-linked dextran beads which are chemically modified to attach to functional groups like DEAE, which impart the specific properties to the resin. A50, it refers to the particle size of the beads. DEAE, diethyl amino ethyl. It refers to the functional group which is attached to Cephadex. DEAE is positively charged at neutral to slightly basic pH due to presence of amino ethyl groups. Thus, it can attach to the negatively charged molecules such as heparinase in our case due to the formation of electrostatic interactions. So now let's discuss the principle of DEAE Sepadex A50 chromatography. First, equilibration. In this process, the Sepadex resin, which is used as the stationary phase, is loaded in the column, and the diethyl amino ethyl groups are attached to it, which gives it a positive charge. Second, loading of the sample. In this process, the sample to be purified is run through the column along with the mobile phase, which is the buffer solution. In this process, the negatively charged molecule, such as in our case, the heparinase is also a negatively charged molecule, attaches to the diethyl amino ethyl groups via electrostatic interaction. Third, washing. Now, after loading the sample, the column is washed with 0.01 molar phosphate buffer to remove the contaminants or the molecules that are weakly attached to the diethyl amino ethyl groups. Fourth, elution. Elution of the target molecules is done by adding a stepwise gradient of NaCl in the range of 0.1 to 1 molar in 0.01 molar phosphate buffer solution. Addition of the NaCl changes the ionic strength of the buffer, which disrupts the electrostatic interaction between the diethyl amino ethyl groups and the negatively charged molecules such as heparinase. Now, different molecules elute out at a different rate depending on their interaction with the diethyl amino ethyl group. The more a molecule is binded tightly with the diethyl amino ethyl group, the more time it takes for its separation. Now, the collected fractions are tested for heparinase activity, and the active fraction molecules, that is the molecules which shows the heparinase activity, are then pooled, concentrated and purified by gel filtration chromatography. After the anion exchange chromatography, gel filtration chromatography, namely Sapphirus 6b column chromatography, is performed. In this chromatography, the Sapphirus 6b resin is used as the stationary phase, whereas the buffer solution is used as the mobile phase. In this type of chromatography, the biomolecules are separated based on their size, charge, or specific affinity for the ligands attached to the resin. The Sapphirus 6b resin is a cross-linked agarose matrix. It is composed of the agarose bead cross-linked with 6% weight by volume agarose. This cross-linking makes the matrix rigid and stabilized, thus allowing it to withstand the pressure and other conditions applied during the chromatography. The Sapphirus 6b resin has a specific force size that determines which molecules would enter the resin and interact with it and which would not. So now let's discuss the steps involved in Sapphirus 6b column chromatography. First, equilibration. In this process, the column is filled with the Sapphirus 6b resin, which is used as a stationary phase, and the mobile phase, that is the buffer solution, is run through the column and eluted outside. Second, loading of the sample. Now the mobile phase along with the sample containing the mixture is run through the column. In this process, the molecules which have size smaller than the beads interact with the resins, whereas the larger molecules are eluted outside. Third, washing. The column is now washed with the same buffer, that is, which contains the 0.2 molar NaCl in 0.01 molar phosphate buffer at a flow rate of 0.5 ml per minute. It results in the elution of different molecules at different rates. Fourth, elution. Now, the molecules which have interacted more strongly with the resin due to their size, charge, or specific affinity move more slowly and get separated afterwards, whereas the molecules with weaker interaction with the resin are separated initially. The eluate is then collected and checked for the heparinase activity. The active fractions are then pooled, concentrated, and its molecular weight is determined using SDS page. After the purification, the active fractions are subjected to molecular weight determination by SDS page. SDS page refers to sodium rhodocyl sulfate polyacrylamide gel electrophoresis. In this process, initially two gels, namely stacking gel and resolving gel, are formed. The resolving gel consists of acrylamide of high concentration, and thus it consists of pore size and thus it consists of very small pore size. The other constituents present in dissolving gel are ammonium persulfate and TMAD, which helps in polymerization of the acrylamide to the polyacrylamide. Ammonium persulfate produces free radical required for polymerization, whereas TMAD helps in stabilization of the free radicals. Also, it consists of SDS, sodium dodecyl sulfate, which converts the secondary and tertiary structure of the protein to its primary structure, that is, a linear structure. It also consists of 1.5 molar tris buffer, which maintains the pH of resolving gel at 8.8. Stacking gel. The stacking gel consists of acrylamide of less concentration and hence bigger pore size as compared to that of resolving gel. The rest of the constituents present in stacking gel are the same, but the only difference is that it consists of 0.5 molar tris buffer, which maintains its pH at 6.8. Now, the running buffer is formed via tris glycine, whose pH is kept at 8.3. Now, after the formation of the stacking gel, resolving gel, and the running buffer, the sample is prepared. For the sample preparation, the tris HCl, whose pH is 6.8, is added to the sample. The tris HCl buffer keeps the pH at 6.8 and also provides the Cl- ion. As it also the sample preparation also consists of SDS, which converts the secondary and tertiary structure of the protein to its primary structure. Then beta mercaptoethanol, which breaks the disulfide bond between the proteins. Also, sample preparation requires the bromophenol blue, which helps in visualizing the position of the protein band in the gel. The sample preparation also requires the glycerol, which helps in maintaining the viscosity required to load the sample in the stacking gel. Now, after loading the resolving gel, stacking gel, running buffer, and the sample in between the two plates, it is covered, and then an electric current is supplied to separate the protein molecules. Now, glycine present in the running buffer plays a very important role in separation of the proteins. Glycine is glycine is a Zwitter ion, that is, it carries both positive and negative charge. At glycine has a isoelectric point of 5.8, that is, it carries no charge at at pH of 5.8. At pH more than 5.8, it starts becoming basic in nature and becomes deprotonated, that is, it carries negative charge. Whereas at pH less than 5.8, it starts becoming protonated, that is, at acidic pH, it carries a positive charge. So, while separating until the stacking gel, whose pH is 6.8, the Cl- ions remains most forward, as it is most negative among the proteins and the glycine. The Cl- ions are followed by SDS protein complex, as SDS gives a uniform negative charge to all the type of proteins. It is then followed by glycine, as glycine is neutral to slightly negative in nature, slightly negative in charge at pH of 6.8. So now all the type of proteins loaded in the sample come at the same point in the stacking gel. That is, in all the proteins, in the forward it is the Cl- ion followed by SDS protein complex and then followed by the glycine molecules. In this way, the protein molecules are not separated based on the charge in the resolving gel. While moving through the resolving gel, the pH changes from 6.8 to 8.8. Due to this change in pH, the glycine molecules becomes more negatively charged. Hence the glycine molecules goes ahead of the SDS protein complex in the band. So now the new band formation is as follows. In the forward, it is the Cl- ion followed by glycine molecules and then followed by SDS protein complex. In the resolving gel, since the acrylamide concentration is high and the pore size is small, the smaller size proteins move faster than the bigger size protein. So the proteins which are the smallest in size are the most forward, followed by intermediate size protein and then followed by large size proteins. Along with loading the sample, a standard marker which consists of protein of known molecular weight is also loaded so as to compare the molecular weight of the unknown protein sample to that of the known protein sample. In this way, the molecular weight of the protein is determined using the SDS page technique. From this chart, it can be seen that using the various purification techniques, the enzyme extract has been purified up to 19 folds. In the present study, the isolation of the heparinase enzyme from Streptomyces variabilis MTCC 1266 and its purification using various purification techniques was studied, and its molecular weight was found to be 42 kilodalton. Thank you for your patience.